So today I'm going to be talking about some details of immune response kinetics and memory immune responses. I will give you sort of a couple of pieces of information beforehand. You will see that some of the stuff at the beginning is going to be similar to things that we've talked about a little bit before, just because we need the segue. I will also say that the number of slides I have for today is considerably more than I can probably do. The big problem that I have for today is that this is a topic that I really, really like and could talk about incessantly and could jump up and down about. And so I apologize for the fact that I'm going to have to hold myself back and that if I start going off on tangents. So basically I have kind of the general overview stuff in the slides first, and then the rest of it are sort of some specific examples of how it plays out in real life with a few different viruses. That's the stuff I may not get all the way through. I'll try to put it in at some other point because it's just really cool. So hopefully we can see all of that as we go forward. So we've seen a lot of the information that's on this slide before. We've seen some type of cell that's an antigen-presenting cell presenting to a T cell, or we've seen a B cell reacting with antigen, those cells being activated, differentiating, and eventually eliminating those antigens. And so all of that is sort of the same general thing we've seen. We also talked a little bit about contraction of the immune response as well. So you can see this same piece of information here where we can look at sort of this general primary immune response. Symptoms are coming up because of pathogen replication and damage coming from that pathogen, which is happening because of the amount of pathogen that is there. The pathogen will be eliminated when we have our maximal primary immune response, and that will stop the symptoms. This on the left is sort of a vague general cartoon version of this. On the right is a figure from your textbook that talks about some of this a little bit more numerically. So here you can see in black the virus replication and sort of what that virus replication looks like over time. In red and in blue, you can see the CD8 and the CD4 positive T cell response. So in both cases, you can see that it takes a little while for those responses to peak. And you can see that those T cells are going to reduce that virus replication level. And I add this figure from your textbook because oftentimes when I show the one on the left, people really want numbers. They want to know what day is the peak day, what day is the contraction day. They want specific days, and so there's some days on this figure from your textbook. I will tell you, however, that the days vary based on which pathogen it is and also the dose of the pathogen, and so that's why a lot of times we don't show the days because they can vary by different things. But here's a version with days if you really wanted them. And we've talked already about sort of this question of what takes so long in order to make an immune response. We need that innate immune response we've learned about. We need all sorts of aspects of the adaptive immune response, the trafficking, the antigen presenting, the activation, the differentiation, movement into germinal centers, movement of cells to the periphery, all of those things in order to eliminate the pathogen. But what we also need to talk a little bit about is that differentiation and more detail about what happens with those cells as they are differentiating. And so when we see some type of T cell that's come out of the thymus or B cell that's come out of the bone marrow, and that cell sees antigen. In the case of T cells, it's MHC plus peptide. In the case of our B cell, it's whatever soluble antigen we have. Those cells will be activated, and we will eventually see two different kinds of T cells and B cells being formed. One type are known as effector cells. The effector cells are all the cells we've already talked about. They have their effect. They are, for example, in the case of a T cell, they might be the CD8 cytotoxic T lymphocyte, the killer T cell. In the case of the CD4, it might be the helper T cell. Those would be the effector cells. In the case of the B cell, it's the plasma cell. We also will see that some of those cells are going to become what are known as memory cells. And part of what we're going to be talking about today is those memory cells. So one of the things that you can notice here is that this figure tells you that effector cells tend to be relatively short-lived, while memory cells tend to be longer-lived. So those effector cells are persisting for a relatively short time and are then going to die, where those memory cells are going to live for a long time. You can see this in that same timing picture that you see on the left. It's the same one I showed you before, in fact. Where it's not like our CD8 T cell response in red goes up to a high level and then stays at that high level forever. We can see that that response contracts and comes down pretty quickly because those effector cells that are making the response are short-lived. They're going to undergo apoptosis. Same for the CD4 cells, although you can see the timing is a little bit different. And that's really, really, really important because if you had that many CD8 cells that persisted forever in response to every viral infection that you had, at this point you would become walking T cells. And you would have no resources and no sort of metabolic products to support any of those T cells in order to make this process happen. And so usually about 95% of the cells that we see during the effector phase of the response, which is the phase where the pathogen is getting eliminated, are going to die in this contraction. This is a programmed cell death that happens. I mentioned before, it's actually programmed into the cells when they first see antigen. It's almost like, in some ways, they're on like a timer of they're not going to live forever. It may be due to lack of antigen, it may be growth factor withdrawal, maybe activation-induced cell death. So we have this activation-induced cell death where the majority of the cells that are present during the effector phase die. And you can see that we do eventually have some number of cells survive due to a whole bunch of survival factors that I'm going to mention. We can easily see the difference between naive and effector cells, and y'all already know this. So if you look at a naive cell, you know that we require signal 1 and signal 2 to turn on that cell. And that's going to turn on the production of a whole lot of different things. If you look at naive cells compared with effector cells have 100 times as much FOS as naive cells. So when they are in the periphery, potentially getting activated, that activation is going to be really easy and really strong. Similarly, you can see they have 50 times more NFAT. They have more MYC. They have more NF-kappa B. They're making way more cytokine, 1,000 times as much IL-2. They're making more than 100 times as much interferon gamma. They're also making way more granzyme B and all of the other products that these cells are going to need. This figure is talking about helper T cells, so it doesn't talk about granzyme B, but it's the same idea. So the effector cells are way better at their job because they have way more transcript and way more ability to translate all of these different products that are helping them do their job. But doing this much transcription and translation of all these products is rather stressful on the cells, and so those cells are going to be the short-lived cells that can go to cell death. And if you remember with the B cell, that plasma cell, which is our effector cell, has actually changed the structure of the cell. It's mostly ER, so that it can pump out lots of antibodies for production. That cell is not going to be able to live for a super long time in that weird state. So these are this sort of shorter-lived effector cells. But a big part of what we've got to talk about today has to do with immune response kinetics. I really like to talk about immune response kinetics. So I want to talk about a few different pieces of what we're seeing here as we go through this. So what you can see is the first time that you are infected with some type of microbe, you will see that nice delay before our adaptive immune response of T cells and antibodies is induced. If you were to get infected with that same pathogen like at the similar time, so let's imagine that you are infected with the flu, and then your sister gets the flu and sneezes on you, like the day after you got better. At that point, your immune response is still going to be relatively high. You might get, the virus might get it into you. It might replicate for like 5 seconds. But your very high immune response is going to wipe it out because it has not yet contracted at this point. And so you could see sort of repeated exposures at that point leading to what are known as abortive infections, where the infection doesn't really progress. It's not really going to sort of alter what we see with the immune response. And these are infections that are happening kind of right after the original infection. So this is we're talking right now about very close in time to the original infection. So this is sort of why, if people in your, in class all have the same thing, you're not going to keep getting the same infection a few times. Once the person next to you gets sick, once you're getting over it and they get sick, you're not going to get sick again. Your immune response is still at a high enough level, it's going to protect you. But what we really need to think about is what might happen with that response later on. So you can see that the response is eventually going to contract to a relatively low level. And if you were to expand this out, you would see kind of this nice contraction, and it would almost go like an asymptote. Like you'd be getting slightly more and slightly more and slightly more contraction over time. Happily, it would still probably always be above this very low level. So at the beginning, you had like 1 T cell that bound to that epitope. You're still going to have somewhat more. So you've got a quantitative increase. But it turns out that not only are these cells better quantitatively, they are also better qualitatively. And so here you can see that second infection, and what you should notice now is that this is way later. It's not like a short amount later, this is like a much longer time later. If you were to get that second infection, you can see that the B cell and T cell response will increase, and it will increase much quicker. You won't have that lag period in the response. And the response will also increase to a much higher level. So this is both a quantitative and a qualitative improvement. You can actually mathematically look and say, well, is the number increase here just because you had a number increase here, or is it actually like an increase in how much they expand? It is actually an increase in how much they expand. I had to calculate that for a paper that I had to do. And so, in fact, it's not just that you started out with more here, so you get more. It's actually they expanded more times. They proliferated more times as well. And so this response that we're seeing is known as a memory response or a secondary response. And here you can see the secondary response. I've had to give short guest lectures in a couple of other classes about vaccines, and like this is the one slide of immunology they get. So here's the slide. So you can see the primary response that I've told you about before. Now you can see a second infection with the same arrow pathogen. You can see that going into that infection, we had a larger number of cells. Those cells are also memory cells, which we're going to talk about more, that are qualitatively better, that are able to respond quickly, and are going to proliferate a lot in order to make this secondary response. And that's going to be so good that it is going to stop that pathogen's replication really quickly to the point where you aren't going to feel sick. You probably won't even know you were infected. And so this is sort of the idea behind you can't get the chickenpox twice. You actually could get it the second time. You could get infected. But the infection is going to be cleared up so quickly by your adaptive immune system that you'll never notice it. You'll never actually see symptoms or start to see the problem. This is now where someone tells me about their cousin who got the chickenpox twice. Huh? Every time. It always happens. So here we can see sort of the general differences between primary and secondary responses. So with the primary response, we started out with a really small number of cells. With the secondary response, we started out with a larger number of cells. With the primary response, there were no antibodies against that antigen present, particularly things that were class switched to IgG or something like that. There actually already are some antibodies present at the secondary response. You can see there's a difference in terms of whether or not we've seen class switch and hypermutation between the primary and secondary response. But there's also a difference in the cells in terms of how much signal those cells need to be activated. In the primary response, those cells need a lot of signal to get turned on. In a secondary response, those cells need a much smaller signal to get turned on. So they have a much lower threshold for a response. In some ways, you can think about it. These cells have already been told, you are useful. You do something. And so now we don't need to control them quite as much. Now they can be turned on much easier. You can see that there's the delay before the T cell response in the primary response. There's no delay in the secondary. In the primary response, the innate response and the adaptive are much more separate time-wise. In a secondary response, the innate and adaptive responses are sort of happening together and oftentimes are quite integrated. This secondary response that we are seeing is largely characterized by the presence of cells that are called memory cells. So the effector cells were the cells that were really important for sort of that primary response and for getting rid of the pathogen during the primary response. But the memory cells are going to be really critical here for this memory response. There are also, this slide is clearly in the wrong place, big differences in terms of the antibody responses between a primary and secondary response. So you can see the frequency of B cells responding to the antigen, the isotypes that we see, affinity maturation, somatic hypermutation has happened. But I was just telling you about how memory cells are special. And memory cells are awesome. So there are a few things about them that are really, really, really important. One of them is how long they live. And so these memory cells are actually programmed to live for a really long time. So these data show some information about half-lives of these cells after infection. In both cases, they're actually looking at after vaccination instead of after infection. And the reason why they do this for these studies is because you can be sure that the person hasn't gotten infected again. So these are actually smallpox vaccines where they do these data. And these are people who got smallpox vaccines right at the end of the smallpox, of when smallpox was present. And so we know this is how long those have lasted since that original vaccination. We know they didn't get smallpox in the middle to mess up this timing. And we can see that if you look, out to 30 years after vaccination, that antibody level is still pretty increased relative to what it was at the beginning. It's not quite as much as at the top, but you can see a pretty high antibody response. The half-life, you can see, is pretty long. The CD4s and CD8s you can see are starting to decline. They maybe have a half-life of 8 to 15 years. But if we actually look, again, after vaccination, we can actually look at vaccinated and unvaccinated people. We see antibodies in the vaccinated people 80 years post-vaccination. And we don't see them ever in the unvaccinated. And if we look at CD4s and CD8 T cells, and we look post-vaccination, you can see that there are CD4 and CD8 T cells, specifically memory cells, out to 75 years after receiving a vaccine. So these cells seem to be extremely long-lived, or to have the ability to proliferate and sort of make daughter cells to keep those cells alive for incredibly long periods of time. These are actually really hard studies to do, because if you imagine, to look at something 75 years after vaccination, you are in a relatively old patient. And largely we can do this with smallpox because, again, that's the one situation where we know you didn't get reinfected. And so we can actually really be looking at the time since you had that initial vaccination. But you can imagine how hard it's going to be to find people who have their smallpox vaccine records, who are 75 years old, who can give us all these data. So there are some people who find these papers sketchy. I think they're great, but I do think that the reason why I'm yeah, they're hard to do, and so yeah, the data's not going to be perfect. That's my feeling on it. So one thing about memory cells that's cool is that memory cells live for a super long time. They're also unique in other types of ways as well. So we can see that there are some proteins that are on the surface of both effector and memory cells. Basically, when the T cell has seen antigen, it starts making that. It's like, I saw antigen, I'm good. And so we can distinguish naive versus activated cells by the amount of, say, this protein called CD44. It's usually known as an activation marker. CD45RO is another one. There are some proteins that only naive cells have, like CD45RA. But what you can see is that the effector cells are going to be a little bit different than the memory cells. The effector cells are going to have lots of granzyme B because they need to be ready to kill right now. The memory cells need to be able to be triggered to make granzyme B easily, but they don't need to be able to kill right now. In fact, that ability to make lots of granzyme B is probably why the effector cells are kind of short-lived. And so here we're going to sort of have some changes. We will see more granzyme B, perhaps, than the naive cell, but we won't see the huge maximum amount. The memory cells will also have some special things, like lots of BCL2, which is involved in cell survival, in order to make those cells survive for a very long time. When we look at, so one other thing that they'll have a lot of is CD127. CD127 is part of the receptor for IL-7. And IL-7 is a really good cytokine that makes cells survive and live. And so these guys are going to soak up all the surviving cytokine so that they can survive for a long time. We can also divide up the memory cells that we see into different subtypes of memory cells. Two of those subtypes are called central memory and effector memory cells. Effector memory cells are closer to being effectors. And it turns out that these two types of cells are keeping a watch for the pathogen, are doing surveillance for the pathogen in different parts of the body. The central memory cells are going to be in the lymphoid organs. The effector memory cells will be in the periphery. And in fact, we can distinguish them based on whether or not they have trafficking markers like L-selectin and CCR7 that allow cells into the lymph node. Central memory cells go to lymph nodes, whereas the effector memory cells do not. And so we can see even further sort of subsetting between all of these cell types. Further, you can see a few other differences between the naive and the memory cells here. So with the naive cells, most of the genes encoding stuff that's important for T cells, most of that DNA is wrapped pretty tightly around histones. The chromatin is closed. The cells don't have a lot of signaling proteins and transcription factors made. So when you start signaling in that cell, the cell has to make all the proteins. And that's part of the reason why there's a delay, why we've got things are so slow. So we need a lot of signaling to get transcription of our necessary genes. In memory cells, the chromatin is open. The cell has been activated. It's been told you're a good cell. And so it leaves that chromatin more open. It also often already has things like TBAT or other transcription factors that it just leaves sitting around. So it's already got its transcription factors. It's already got signaling proteins. It's already got the DNA that it needs kind of accessible. And so what that means is it doesn't take very much to stimulate that cell. That cell, as soon as it gets a little bit of a signal, it's ready to go. That's why you can see that cell respond so quickly as opposed to before where we needed a little bit more time to make some of these transcription factors. This is also why we can see such a strong amount of proliferation, because these cells are sort of predisposed to be ready to proliferate. So these cells are going to be producing tons of cytokines. They are great cytokine producers. They also do not need signal 2 anymore. So signal 2 is really important for turning on a T cell the first time, but once that T cell has been turned on, it doesn't need signal 2 anymore. So these cells, again, are sort of like really ready to go. They're just waiting for a little bit of signal to tell them it's time to go again. Whereas the naive cell takes a little bit more work in order to turn that cell on. So there's one thing that I have skirted around in talking about all of this. And the reason I've kind of skirted around that is because it's somewhat controversial in the field. And so let me give you, for example, this slide. You see that there's naive cells, and they become effector cells, and they become memory cells. But what I didn't really talk about is how are effector cells and memory cells related? This kind of tells you that, like, this figure tries to have their cake and eat it too. It gives you both models on this figure. So some people are like, no, effector cells turn into memory cells. And some people are like, no, naive cells turn into memory cells. And there's a lot of argument about which one of those things it could be. What you could imagine for the people who say effector cells turn into memory cells, basically what they're saying is the ones that live through the sad time of contraction just magically get to be memory cells. Hooray! That's basically what that model is saying, is that if you live through this, if you're an effector cell that's still alive here, you turn into a memory cell. The other idea is that when those cells are originally turned on, some of them are like, you are going to grow up to be an effector cell, do all the effector cell things. You are going to grow up to be a memory cell, do all the memory cell things. And so the effector cells are the ones that died, and the ones that were told to grow up to be a memory cell are the only ones that are still around. And so those are kind of the two models that people have. So some people think that the memory cells are just the ones that lived through being effector cells. Other people think that a naive cell can either turn into a memory cell or an effector cell. It has a choice. There is some pretty nice data for both of these models. I remember when I was working on my dissertation, I found this paper. It's actually the paper that kind of leads to this figure. I found this paper, and I loved that paper. Oh, it was the best. It told me all about how memory cells are formed. My favorite paper ever. And then I kept turning the pages of the issue of the journal, and the next one after it supported the other model really well and was like this gorgeous figure. So like people go totally back and forth on this one. So there's this super cool paper that I love that says that when the naive cell originally interacts with the APC, it moves its cytoskeleton and divides in this direction. So it ends up with a close daughter cell and a far daughter cell. and the close daughter cell becomes the effector, and the far daughter cell becomes the memory cell. Yeah, like that paper is the coolest paper. I love that paper. What do you mean? So what they did is they did a whole bunch of microscopy and saw things like this. And what they realized is that these two cells, as these two cells were dividing, there was one protein that got partitioned differently between them. There is a way you can look at cell divisions by flow cytometry, and so they did cell sorting on cells that had undergone one cell division and that had which different proteins, so they could actually get the close or the far ones, and then they grew them in culture and watched them grow out to the two different things. Or they transferred them into mice and watched them go into the two different things. It's really cool. And their follow-up paper is even really cooler, but I can't. Yeah. It's entirely possible so people are coming up with other types of models, and it may be some of that as well. One of the things that I think people are really talking about now is something that's a little bit more like this, which is sort of a more progressive model. So if a cell gets sort of a weakish signal, it becomes a memory cell. And if it gets a stronger signal, it becomes an effector cell. And so like every cell starts as a memory cell, and if they keep getting pushed, they end up as an effector. And so like the idea in this is actually, so the idea is that all the cells start out as memory cells. Some of them get pushed really far to effectors, and some of them don't get pushed as far and stay as memory cells. Those memory cells are almost like stem cells that can replenish the response to that type of thing. And so this is the stem memory model. And so there are, again, sort of other ways that people are thinking about this process. But people always want to ask that question of where the memory cells come from. And my honest answer is, we don't really know. This is kind of the best we got. So a couple of other things just to say. Like I said, we can easily detect different types of T cells, whether they're naive, whether they're effector, or whether they're memory cells, based on flow cytometry markers. I used to get in fights with my PhD advisor about this pretty often, because when I was in grad school, there were naive, effector, central memory, and effector memory. Those were the four. And sometimes how you did effector memory and central memory looked different in different experiments. And so we were talking about why that is. And I would always be like, I think this is dumb. I think there's more than just those kinds of memory cells. I think there's more than two. And that's why experiments come out differently when you do them with different flow cytometry things. And he's like, no, I think it makes sense that there's four. And I was like, no, there's only four because you did this with your flow cytometry plot. And so you called them four things. Like, if you did your flow cytometry a different way, you get more than four. And so we argued about that. And so sometimes when I see things like this, where they show lots of kinds of memory cells, I feel vindicated. And one thing that you can also see is if you look back at the Bura paper that we talked about last week, where they were doing some of this memory differentiation, the markers that people tend to use for mouse work and the markers that people tend to use for human work are slightly different, which is, again, one of the reasons why it's hard to work some of these details out. A couple other sort of general pieces of information. One to know is, especially for CD8 memory cells, this is also true for memory B cells, CD4s are really important. So you actually can't get a good CD8 memory response without a good CD4 response. These figures are actually also putting together a bunch of data that came from some really important, cool papers. I will point out, there's a reason for this. One of the authors of one of these papers was a grad student, like the first author of one of these papers. I can't remember which of the two it was. I used to definitely know, but I can't remember. It's one of these two papers. The first author was a guy named Joseph Sun. I'm telling you that because in the lecture we talk about NK cells, Joseph Sun comes up again. So it's like you're basically going to learn about all of the things Joseph Sun did this week. So one of these was Joseph Sun, but I can't remember which one it was. But basically what they did was, what was done in these two papers was they took a microbe, here it was a bacterium called Listeria monocytogenes, that had a specific antigen that is expressed on T cells, or that is recognized by certain T cells called OVA. They looked at mice that were wild type or mice that were missing MHC class 2. And remember that mice that are missing MHC class 2 will not have CD4 cells, because CD4 goes with MHC class 2. And they looked at their CD8 responses at 7 days after infection, and they saw they were exactly the same. But then if they looked 70 days later and tried to reinfect the mice, the ones that didn't have CD4 cells couldn't make a secondary response. And so it looked like those CD4s were really important for helping CD8s make their memory response. Similarly, they infected mice with a virus and then put CD8 cells either into a wild type or a class 2 deficient mouse, singular plural, and all of the CD8 cells, when they were in mice that didn't have CD4s, they all died. And when those CD8 cells were in normal mice, they lived. And so it looks like there's a lot going on here in terms of interactions between different cell types in order to make this whole process work. One way that CD4 T cells might be involved in thinking about or in helping with CD8s is through cytokine production. And so in order to make, say, for example, a really good memory CD8 response, lots of different cytokines are needed to support those cells in becoming memory cells. So you can see there are some cytokines like IL-2 that are really important in the effector phase. Maybe IL-15 really helps those CD8s become effectors. But you can see that other cells are really important for helping those memory cells sort of stick around long term as memory cells and sort of hang out for maintenance. And so it looks like a lot of this process of helping cells go into being either effector cells or memory cells is based on cytokine control. To give you one example of what that looks like, I promise this is not just self-promotion. So this is a figure from my dissertation. Well, it's a figure from a paper, but it's also in my dissertation. And so here I was looking at normal mice, and the normal mice are in purple, wild type mice. And I was also looking at mice that were missing a receptor for a cytokine. So specifically, they were missing the alpha chain of the IL-21 receptor. So IL-21 receptor alpha deficient mice. And I was infecting them with a particular virus, and then measuring the virus-specific cells in their blood. And first, if you just look at the purple line, this is sort of the primary response to the virus that I saw. And then at day 56, 8 weeks later, I reinfected them, and you can see my nice secondary response that I saw. When I looked at my knockout mice, they actually made a response that was pretty similar at peak to the primary. The cells died a bit more easily, but when I reinfected those mice 8 weeks later, they made another primary response instead of making a secondary response. So in fact, the peak here and the peak here were identical. And so you can see that cytokines can actually really control what's going on with these cells and whether or not they are able to become memory cells or effector types of cells and make the responses that we're talking about. Okay. So are we good right now? Okay. So everything else is sort of like examples of stuff. The first one is a relatively long example. It's also an example that most immunologists find it's pretty famous. Lots of people talk about it all the time. It's also one that non-immunologists find baffling. So I'm going to try to go through it so that you totally get it and know what's going on, and then you can, like lord it all over all your friends that you understand it. Because I'm sure this is what you talk about all the time. And this particular issue, in this particular situation, all has to do or comes down to things that are happening because of what we see here in these immune response kinetics. So I want to talk a little bit about a response to a particular virus. I'm going to flip around the order of a couple of these for a second. So I'm going to be saying this, that it's influenza. It doesn't have to be influenza, but everybody always uses flu for this example, so it's going to be flu. This is flu. There are two big things you need to know about flu for this example. One of them is that flu undergoes a lot of types of genetic change. So we're going to see a lot of changes in flu viruses between different years because of some of the unique aspects of the RNA genome of flu. So one issue is going to be lots of genetic change. Basically assume every year's flu virus is different than the flu virus before because of these issues of genetic change. There are 8 different proteins in flu that are important, but only 2 of them are we ever going to care about here. They are the ones that are kind of these flowery looking ones on the outside, which are known as HA and NA. HA and NA are the things that let the virus into the cell. They're also the things that you tend to make antibodies against when you make antibodies against the flu. So you now know everything you need to know about the flu. Fun fact, you might have heard of flu being called like H1N1. That means it uses the number 1 kind of HA and the number 1 kind of NA. And if it's H5N1, it means it uses the number 5 kind of NA and the number 1 kind of yeah, anyway. So that's where all this naming comes from. So this process is something that can happen because of memory responses. And you might look at this and might realize there's some problems here. So let's imagine we have this person who got infected with this flu strain. It's the strain that's shown here. It contains 4 epitopes, A, B, C, and D. So our person is going to get infected with this flu strain. They are going to make a primary response to antigen A. They are going to make a primary response to antigen B. They're going to make a primary response to antigen C. They're going to make a primary response to antigen D. It's going to take a little while before they make that nice primary response. And they are going to feel terrible because the flu is replicating during that time. They had no previous immunity to this flu virus. So they are sick in bed for 14 days. Now let's imagine that person gets the flu again the next year. The flu virus has undergone some genetic change. And so the flu virus has changed so that it now has epitope A, epitope B, epitope C, and epitope E. What kind of responses, what's this person going to make in response here? Yeah, Nick? To what? Yeah, so this person's going to make a secondary response to A, a secondary response to B, a secondary response to D, or sorry, to C, okay? Those responses are going to be really fast and really big because they're secondary responses. And those are going to get that virus gone really fast. And that's a good thing. Oops. That's a good thing. Look, the person's not really going to get sick in bed. They're not going to notice they're sick. I like the 14 here, and I like the 14 here. I don't love the numbers in the middle. So for all of these, we can kind of say not sick. So they're going to make this really nice memory response. They're never going to know they got the flu. They're going to feel fine. What kind of response are they going to make to epitope E?
They would make a primary response if that response weren't being shut down by all of the secondary responses. That, you know how you said that they stop for one from
Okay, what were you going to say?
I was going to say the thing about the response to it due to the fact that they're already having.
Yeah. So they're actually going to not make a response to E. So you can see no response to E here. There are two reasons for that. One of them is something that we talked about when we talked about B cells before, that the activated B cells shut off naive B cell responses. So the B cell responses are going to be actively turned off. The T cell responses aren't going to be actively turned off, we don't think, but this virus is just going to get cleared so fast, there isn't going to be enough time to make a T cell response. So it's more about the timing. The virus is just gone super fast, so the T cells don't even get a turn. Like the naive T cells don't even get a turn to get activated. So you're making nothing in response to E. So now let's imagine this person gets the flu again. They get, and the flu virus has undergone genetic change. It has now antigen A, antigen B, antigen E, and antigen F. Oh wait, hold on a second. Look, this is the thing I told you before. The memory B cells turn off the naive B cells because of having pre-existing antibodies signal through that negative signaling molecule to turn this thing off. So this is actually the current version of this in your textbook with the negative signal on B cells. All right, so what happens the third time around? Here. Yeah, Mark. So it's not, so almost. So in reality, what happens is you have a response to A and B that is officially called a tertiary response, because it's the third time. And you know what happens in a tertiary response? Primary, secondary, tertiary. I already ran out of board. That's sad. It actually keeps getting bigger and keeps going up again. So now they're making massive tertiary responses to A and B. And those are swamping out E and F. We're getting rid of that virus so fast. We're turning off B cells and we're getting rid of that virus so fast, we never make a response to E or F. Okay, now what happens the next year when they get infected with a virus that has A, G, E, and F? Yeah. They make a quaternary response to A, so they make a whopping response. I don't have enough board to show you how big their response would be. But they make so much response to A. Again, we get rid of that virus real fast. And there's never a chance to make a response to E, F, or G. So now what happens in year 5? Okay, how about if we don't die? Yeah, Vanessa.
It's almost like at the there's nothing to respond to, so it has to go back to the inflamed.
Yeah, we're now going to see a primary response to these antigens E, F, G, and H, even though this is the 4th time you saw E. This is the 3rd time you saw F. This is the 2nd time you saw G. You're still going to make primary responses to all of them. You're still going to feel really sick. It's still not going to work. And so what you can see here is that your previous history of antigen exposure makes a big difference in what happens to you after your infection. So you could imagine that the same pathogen is going to be giving slightly different types of disease to people based on what they have had a previous infection with. This phenomenon, where your original infection, the original pathogen that infects you, messes with later responses that you make, is known to immunologists as original antigenic sin. And so this is the process of original antigenic sin. A lot of the most famous examples of this happen with flu. And I already told you some stuff about flu. So, what? sin. Yeah, like original sin, like in religion, except it's original antigenic sin. See, nobody laughed before. I was a little worried. I guess nobody was understanding the word I was saying. So this actually will tell us, it turns out that if you look at flu, we see different types of flu viruses that are sort of predominating at different times. So before 1918, one called H3N8 was predominant, and then H1N1, and then H2N2, and then H3N2. There's some H1N1 that came out later. And so the reason why I'm showing you this is that if we think about, say, the United States population right now, different groups of people, different age groups, had a different kind of flu be their first flu, be their original antigen. There are a few flu viruses that people tend to worry about. Some of you might remember swine flu, which happened in 2009. This is H1N1. It was not fun. I did not enjoy it. It was estimated it would have infected 1 in 6 Americans in 2009. Fortunately, only 0.26% of people who have it die, but you feel pretty bad. And in fact, it was first detected in April. It was June. The vaccine was out in October, and the pandemic was over by the following year. But it was quite problematic when it happened. You may not remember there was another type of flu called H5N1 or bird flu. It is still in circulation today. In fact, there have been a number of cases of it in Egypt somewhat recently. This one's really scary because the percent of people that die when they get it is 60%. But it doesn't really transmit very well from person to person. There have only been 860 cases total. This slide's a little old, but we're certainly not over 1,000. So these are two flu strains that a lot of virologists spend a lot of time thinking about in terms of whether they could lead to a major pandemic. And you can imagine this one would be real bad, because if it led to some major pandemic, 60% of people who get it die. There's also one that is has been seen more recently called H7N9, which was first detected in 2013. 39% of people who get this one die. And there's a lot of evidence that this one potentially could end up moving towards being a big pandemic. So it turns out that what has been shown is that whatever your first influenza was, whatever type of flu you had first, influences how susceptible you are to these. So it seems as though if your first flu was one of these kinds, group 1, you seem to be pretty well protected against these other group 1s, and you seem to be out of luck against group 2. And if your first flu is a group 2, you seem to be in pretty good shape against group 2 and be out of luck against group 1. What that event actually seems to mean, we spend a lot of time now thinking about the ghost of influenza past, is it turns out that depending on what flu you had as your first flu, there's a big difference in what's going to happen. And so just to sort of show it, one of the first things people realized was in 2009, way few people died of swine flu. Like, usually a lot of elderly people die of flu, and it was very few people. That, like, way fewer than you would have expected. And some of that's because it turns out their first flu was a similar H1N1. So they were actually protected in a way that other people kind of weren't. It also seems as though people who were born in this time period before 1968 probably had their first flu was either H1N1 or H2N2. Those are really similar to H5N1, and so when we look, they're actually really protected against H5N1, that bird flu. They might be fine. if there was a bird flu epidemic. However, they make awful responses to H7N9. And so if there's an H7N9 pandemic, they are particularly vulnerable. People born after 1968, their first flu was probably an H3N2. And it turns out H3N2 is really similar to H7N9, and people born after 1968 are probably going to be in better shape if we have an H7N9 pandemic. But if we have an H1 or H5N1 pandemic, those people are out of luck because this first flu is leading them to a different response. And so some of what we see sort of population-wide is based on what we see with pre-existing immunity and the impact of memory responses. And so this is sort of what science had to say about this, that we can see big differences in whether we would expect to see, we'd see mostly H5 cases in a younger population. And so if we had a flu epidemic where there was H5 being predominant, mostly it would be these people who were really sick and having problems. If we had an H7, it would be mostly these people who were really sick. And so all of this can be figured out based on some details of the original infecting antigen. The other thing that sort of plays into this is this issue of cross-reactivity. So I have always kind of told you that one T cell responds to one antigen, one B cell responds to one antigen, one epitope. But unfortunately, biochemistry is annoying. Sometimes closely related pathogens can have very similar antigens or epitopes. So here you can see two viruses that are closely related, cowpox and smallpox. And you can see that they have some antigens, like the red square and the purple cross, that are unique. But they also have some antigens that are shared because they're so closely related. As a result, if you are infected with cowpox, some of the antibodies you make in response to cowpox will actually bind to and potentially protect you against smallpox. This is a good thing because the smallpox vaccine was based on a similar pox virus. We now think it's horsepox and not cowpox. In any case, it still has some similar antigens. And the idea is that we're using that cross-reactivity by giving you a virus that's not going to cause you harm, inducing you to make those antibodies, and knowing that those antibodies are going to cross-react. The bad part of this is what this also means if you put, and in fact, this was first done by Edward Jenner. We'll talk more about Jenner later. I don't need to talk about Jenner now. But there's a problem with all of this as well, because what this should tell you is that those original antigenic sin things, those things where an earlier infection influences your later infections, won't just be between type of flu number 1 and type of flu number 2. Whether or not you've been infected with a cross-reactive pathogen can also influence later immune responses. And so here we saw investigators who were infecting mice with two different microbes. One is a virus called LCMV, one is a virus called PV. And weirdly enough, if you actually look at the amino acid sequence in some of the proteins, they have almost the same protein, basically almost the same epitope. And so if you look at a mouse that's infected with LCMV, you can see that that mouse will make some CD8 positive, interferon gamma positive responses. Those are good effector T cell responses to 4 different LCMV antigens. You can see that it makes the most of NP396. And it turns out NP396 is really good at killing LCMV. So those mice feel happy after their LCMV. If the mouse previously had this other virus, PV, it changes which epitopes those mice are making responses to. Now, those mice make whopping responses to one called NP205 because of its similarity to a PV epitope. And so now, these mice are making a totally different response to this virus. The mice have the same genetic background. It's the same virus. The only difference is the previous antigen exposure of the mouse. And so this difference in which epitope is targeted can lead to a difference in the outcome of these different mice. The next slide is identical to this slide and is apparently a duplicate. And so, but anyway, on the side over here, there's a graph of how much of the response is directed towards each epitope, and you can see that when they got just LCMV and when they got PV and then LCMV, there was a huge shift in what the responses looked like. And this is sort of just another way of showing that. So here are mice that got LCMV. Here are mice that got PV, and you can see that there are sort of huge changes in what types of responses they're making. So the point of me telling you all of this is that this is one of the reasons why, if I were to be some sort of evil henchman, and if I brought in some pathogen and infected all of you with it, you would all have a different disease. Some of you would be totally fine. Some of you would get really sick. One of the reasons for that is because of your previous infection history. Because you all have a different previous infection history, some of you will have cross-reactive memory responses, some of you won't. And so it will be a whole sort of mismatch, or not mismatch, but a whole mess of what kind of responses you make. If you add to that the fact that you all have different MHC types and things like that, we can start to see immense variation in what happens to people even if they are infected with the same pathogen. And so in the real world, everybody who's infected with something doesn't get the same disease state. There's a lot of variation for a lot of reasons. This is one of those reasons. The other examples that I wanted to tell you about are actually relevant to vaccines as well. And so I may put them in with vaccines, or I may put them in some other time because they're important. One in particular is very important. So I will see you guys in lab tomorrow. Make sure that you at least take a look at your data, and you should have time to analyze it tomorrow in lab if you want to do so. We're also going to finish the ELISA protocol tomorrow in lab, and I will see you then.